Hello, future doctors. Welcome back to Best Medicine's AQA GCSE Biology series for students in the UK. Today, we're going to be talking about the DNA, its structure in detail, genes, and how genes are converted into proteins. And also, what are mutations? So, let's start from the very beginning, right? Remember that we have a cell, right? We're all made up of cells. And so, because we're made up of cells, we are eukaryotic organisms. And in our nucleus, we have the DNA, right? That's something called the DNA. So what is the DNA? DNA essentially is a molecule that codes for proteins. So what it does essentially is that it gives out a little bit of a signal, a template, and that template is going to go to the ribosomes, travels outside, and outside we have the ribosomes. And then those ribosomes are able to read off that template, translate it, right? Able to understand what it means, and then make a protein. And so it goes ahead and makes a protein. So that's the DNA. What does the DNA consist of? What does it look like in its details? That is in your syllabus, and so we have to understand, we have to discuss how it works and what it looks like. Now, the structure of DNA essentially is a double helix. Let me show you again. Over here, right? DNA kind of looks like this. You've seen this in movies and cartoons and hopefully one of our videos. And it's got two strands, right, as you can see. And then on top of this, it's got these twists and turns. And so because it's got these twists and turns, we describe it as having, number one, two strands, right? Number two, helix. So two things. It is a double-stranded molecule that is helical in shape. Double-stranded molecule that is helical in shape. And that is 2 marks for a question that will hopefully come up about the structure of DNA. So DNA is a polymer, is a biological polymer that is made up of 2 strands, which is helical in shape. Now, on a structure, it contains genes that code for proteins, which as a result perform various functions. So therefore, the genes, they give out a template. Template is translated by the ribosomes, converted into proteins, and proteins go ahead and perform different functions. So this information comes from genes. For example, you could have a gene for insulin, right? So you could have a gene for insulin that's going to go into the ribosome. The ribosome makes the insulin, and so on. So for example, a gene for insulin makes insulin protein, which performs the function of decreasing blood sugar in, for example, when it targets the liver cells and the muscle cells and so on. So how does the structure of DNA look like? We already discussed this on the right side. So in a human cell, DNA exists as a thread-like structure called chromatin. However, just before cell division, DNA gets supercoiled to form chromosomes. This is something that we already finished, but let me just quickly show you. Basically, what occurs is that DNA normally, right, in its entirety as a polymer, it kind of looks like this. So that's a double-stranded helical structure. But then, this is wrapped around some proteins. So this is essentially wrapped around some proteins, kind of like beads on a string. So here's a bead, here's a bead, here's a bead. And here's your DNA coming in and wrapping around these, okay? And that is forming this long cylindrical thread. That thread is called chromatin. So DNA normally exists as chromatin, but then this chromatin is going to condense to form chromosomes. So this chromatin eventually is going to condense, right, to form chromosomes just before the stage 2, the M phase of the cell cycle. All of this has been previously discussed, so I'm going at this in a very fast pace because I expect you guys to have watched the previous videos. Okay. Now, in humans, when DNA gets supercoiled, it forms chromosomes, and we're able to see 23 pairs of them. So we have a total of 46 chromosomes, 23 pairs. And each pair contains 2 chromatids, right? So for example, this is pair number 1, and then here's chromatid 1, and then here's the other chromatid. And we have 2 chromatids. Actually, one of each of these come from each parent. So this could be from the male parent, and this could be from the female parent, right? Now, the chromosomes 1 to 22 are called autosomes, right? And the last chromosome, chromosome 23, is called what? It's called the sex chromosomes. Now, sex chromosomes can be of 2 types, X or Y. If you have 2 X's, that's a girl. If you have an X and a Y, then that's a boy, right? That's a male. What is a genome? A genome is a word that often shows up. This is what this chapter is about as well. So what is a genome? A genome stands for all the genetic material inside an organism, right? The entire genetic material combined, containing all the genes, all the coding regions, all the genetic regions, as well as the non-genetic regions of the DNA. So the entire set of DNA in a human cell is the human genome, okay? Because all cells have the same DNA, so just one particular cell's DNA is the human genome, okay? The human genome contains around 20,000 genes. We have actually 20,000 genes. What is the structure of DNA on a molecular level? That's what we want to do, right? That's very, very important. That's something that comes up, and that will help us understand how DNA is able to code for proteins, right? How is DNA able to code for proteins? So remember, the function of DNA is to code for proteins, and therefore we have to understand its structure. Now remember, DNA is a biological polymer that is double-stranded and helical. Because it's a polymer, it must have monomers, right? So because DNA is a polymer, it must have monomers. And the monomers of DNA are called DNA nucleotides, okay? Nucleotides. DNA nucleotides. DNA nucleotides are composed of three things, and you have to know these, okay? DNA nucleotides are made up of, number one, they have a phosphate. Number two, they have something called a ribose sugar. A ribose sugar. Deoxyribose sugar, right? Deoxyribose sugar. Deoxyribose sugar. And that's a 5-carbon sugar, as you can see. We also call it a pentose sugar, right? Just like a glucose is a hexose because it's got 6 carbons. We also call it a pentose sugar. So deoxyribose sugar, you can, for GCSE exams, for AQA at least, you can just write sugar, and that's enough. So we have a phosphate. We call it a phosphate group. Phosphate. It's an O, phosphate group. A deoxyribose sugar, and I expect you guys to know how to draw this, so I'm going to teach it to you again. The way to draw this again is to draw an oxygen and draw a pentagon around it. Why a pentagon? Because it's a 5-carbon sugar. So here's carbon number 1, carbon number 2, carbon number 3, carbon number 4, and the 5th carbon is actually at the side chain, so carbon number 5. And this 5th carbon is going to attach to the phosphate. We'll talk about that in a bit. The third thing, the final thing that is present in a DNA nucleotide is something called a nitrogenous base, right? A nitrogen base. And these could be any from adenine, thymine, cytosine, or guanine, okay? Adenine, thymine, cytosine, and guanine. While in RNA, which is another particular molecule, I want to add this a little bit. So in RNA, which is the template that we make from DNA, there's something called an RNA, ribonucleic acid. DNA is called deoxyribonucleic acid. We also have RNA. So RNA is a ribonucleic acid. It contains, instead of thymine, it contains uracil, okay, which is dictated by a U. And each of these is dictated by an A, T, C, or G, okay? Now, these bases are further classified into categories, and I expect you to remember these categories. So, bases can be divided into purines. Purines are the big ones, really. So, purines is very easy to remember. Purines when you know the pyrimidines. So, pyrimidines you can remember by the mnemonic CUT cut, okay? CUT cut. So basically, cytosine, uracil, and thymine are going to be pyrimidines. So cytosine, uracil, and thymine are going to be pyrimidines, which means the rest of them are going to be purines, and that's going to be adenine and guanine. Now there's something called complementary base pairing. Complementary base pairing is that if you bring two bases in front of each other, they will form weak bonds between each other, okay? They hold each other by bonds, and that is called complementary base pairing. and that is specific to specific pairs of bases. So the way this works is that in DNA, adenine will always bind to thymine, okay? Adenine always binds to thymine, and guanine always binds to cytosine. In RNA, because we don't have a thymine, we have a uracil, then in RNA, adenine will bind to uracil. Okay, very simple. I know it looks a little bit complicated, but it will be broken down, and you'll understand it much better in a very short while, okay? So bear with me here. Nitrogen base. So over here we have the base. So this could either be adenine, thymine, cytosine, any of those. Alright, so what happens is that we form a bond between the phosphate and the 5-carbon sugar, right? The pentose sugar, the deoxyribose sugar. And on the other side, we form a bond between the base and the sugar. And that is our DNA nucleotide, our monomer for the polymer that's going to be DNA. So now what we do is we form a strand. Let's try to form a strand out of this. Okay, so here are a couple of bases, just like this, in which we're going to have different kinds of nitrogenous bases. So this is what a base looks like, remember. This is what a DNA nucleotide looks like. Every DNA nucleotide contains a phosphate group, a deoxyribose sugar, and a nitrogenous base, a nitrogen base. Now, what these DNA nucleotides do is that they go ahead and link with each other to form a single DNA strand. So let's have a look. The first nitrogenous base, maybe it contains adenine here, right? So the first nucleotide contains a nitrogenous base that has adenine in it. The second, maybe it contains thymine, right? The third, maybe it contains something like guanine, right? Over here, let's make a C, for example. So the way they get linked is that we have the phosphate that forms a bond between this carbon here, this carbon there, okay? In our orientation, that is the third carbon with which the phosphate forms a bond there. This bond, you're not supposed to know it for AQA, but I will give you the name anyways. It's called a phosphodiester bond. Diester bond. Phosphodiester bond. If you know ester bonds from organic chemistry, then you should be familiar a little bit about what an ester bond is. But all you need to know essentially is just remember this bond name if you want to remember the name. But for AQA, I don't think you need to even remember the name. So you'll have all these phosphate bonds between adjacent DNA nucleotides, forming a single strand of DNA molecule, right? So you have all these phosphodiester bonds, and we formed a single DNA strand. And what's going to occur is that in front of it, you'll have an anti-parallel adjacent strand, right? Anti-parallel strand that is complementary to the bases that are present in this particular strand. And remember, the bases undergo complementary base pairing, adenine always to thymine and guanine always to cytosine. So let's draw a nucleotide here. Here's a nucleotide, and because it's going to be complementary, it's going to be upside down, right? Anti-parallel. And you're going to have a phosphate here. And there we go. So we get an anti-parallel strand that will have complementary bases in front of the first strand. So adenine always binds to thymine in DNA, right? That's a DNA strand on the left, a DNA strand on the right. So adenine will bind to thymine. Thymine to adenine, of course. Guanine to cytosine, and cytosine to guanine, and so on. And so we have these two strands, and they have these weak bonds in between them, and we get the two strands. Now, what I'm going to do is I'm going to go ahead and draw these strands in a very simple way, okay? Drawing these two strands, and here's the first nucleotide, so that's adenine, okay? I hope you guys are following. So this is here, and then the second one is here. That's thymine, and then the third one and the fourth one. So thymine, we've got guanine, we've got cytosine, and then on the other side we have thymine, adenine, cytosine, and guanine. Okay? And we have this particular order, so I can even keep on going with this, remove the bases from my drawing, and just make this ladder structure. So as soon as this particular double-stranded structure is formed, this is just going to naturally go ahead and fold onto itself. Or not really fold, but really curl itself up. So what it does is that it twists and turns and forms a helix. Okay? Naturally. So that is your DNA. So DNA contains two strands that are anti-parallel. So if this is going up, actually, if this is going down, then this is going up. Okay? And if this is going down, then that one is going up. In this particular case, for using A-levels knowledge, that's a 3 prime, so this is going to be going down from 3 to 5. That's going to be going up in this particular order for this particular strand. But again, you don't need to know order. So all you need to know is that DNA is a double helical structure, double-stranded and helical structure, and that it contains these three things: phosphate, ribose, and nitrogenous bases, complementarily attached to each other. And the DNA nucleotides are linked together by phosphodiester linkages, forming something called a phosphate backbone. Okay, that's another term that we need to know. A phosphate backbone. So let's write that here. The backbone that is linking the entire DNA nucleotides in each of the strands is called a phosphate backbone. A phosphate backbone. Okay? Very simple. Now, a question arises as to how do we go from this particular thing here, right, to entire protein, right? How do we go from this to a protein? And that needs a little bit of an explanation. So let's have a look. Remember, in a nucleus of a cell, let's make a big nucleus here. So here's a nucleus. In the cell, you have the DNA, right? Here's the DNA. I've opened this up a little bit. So here's the DNA. Now, what the DNA contains is that it contains these bases. For example, there's T, right? There's A, there's C. I could make like T here, T here, T here. Okay, and then you would have complementary bases in front of it, and so on. Let's say that this is part of a gene, and we want to make a protein out of it. So this is a gene, and we want to make a protein out of these bases. Well, the bases are able to help code, actually, for a sequence of amino acids. The bases actually tell us what amino acids to put and when. How do they do this? Here's how it works. The bases code into a template, a messenger that is going to travel to the ribosome and deliver the message of the order of amino acids to be put together. Let me repeat this. These bases on the DNA are coding for a sequence of amino acids that is carried by a template to the ribosome that will link the amino acids together in that particular order. So how does this work? Well, this is transcribed. We call it transcribed because it's converting the same information from one form to another. Just like audio to text is transcription, over here DNA to RNA. An RNA template is formed. An RNA template that is going to take the message from the DNA to the ribosome. This RNA template, because it's taking the message, is called a messenger RNA. So messenger RNA is going to take the message of the sequence of bases to the ribosome. How does this work? Well, complementarily, mRNA will be formed. So mRNA is going to have an adenine here, because thymine always binds to adenine, and a uracil here, because adenine binds to uracil instead of thymine on the mRNA. mRNA does not have thymine, so we're going to have a uracil here and a guanine here. And for the rest of it, it's going to be A here, another A, right? For example. Now, this template that we have formed, this messenger RNA, containing AUG, AAA in our example, right? That's going to go to the ribosome. So let's talk about what happens at the ribosome. Maybe make this diagram a little bit bigger. Okay. So here's the ribosome. And here is our mRNA, messenger RNA, which is bringing in the sequence of bases that codes for the sequence of amino acids. Now here's the thing. Every 3 bases together are going to code for one amino acid. What I mean by this is that these three, right, this triplet here, this triplet is going to code for one amino acid, and then this triplet is going to code for the next amino acid, and so on. Each of these triplets, right, every triplet on the mRNA is called a mRNA triplet, is called a codon, okay? It's called a codon. So if one triplet, right, 3 bases are coding for one amino acid, then maybe 30 codons, or 30 bases, right, will code for 10 amino acids, because 3 of these code for one amino acid. How does the coding work? Let me show you. So because each of these are going to code for an amino acid, we have to have a system to recognize the amino acids in the cell to come and bind or come to the ribosome when these bases come here. And we do have a system. So the system essentially contains something called transfer molecules or transfer RNA. Here's how it works. Let's say here's an amino acid, right? There's an amino acid called methionine, so I'm just going to write it here, right? Methionine. And what you will have underneath methionine is something called a transfer RNA, right? An RNA that helps transfer the amino acid to the ribosome. This transfer RNA is going to have RNA that is complementary to the triplet, to the codons, okay? So this transfer RNA is going to have complementary RNA here on it that is complementary to the codons on the mRNA. So for example, we have A here is going to have a U here because it's an RNA, right? This is called a transfer RNA. And because we have a U here, so that's going to have an A here, and we have a G here, so it's going to have a C here. And that's how the ribosome will know what amino acid I want to put. So AUG is going to code for methionine, and it's going to come in and link up here. So what shortly follows is that as soon as AUG comes to the ribosome, for example, then the transfer RNA containing the amino acid methionine is going to come in and pair up here, okay? And binds to, using the complementary bases, UAC, binds in front of the mRNA. And now it brings in with it the amino acid, right? Methionine. Now what's going to happen is it will drop off methionine and leave. So it's going to go ahead and leave now. Empty, right? Look, it looks kind of weird, right? Clubber shaped. But it's just going to leave. So now we have methionine. Let's just go ahead and remove this area here. So what we have now is methionine. I'm going to put methionine to the side, or maybe we can make another diagram. So shortly what follows is that, so we have the ribosome and we have the methionine already there. Now the RNA has moved along, right? AUG is here because it's already deposited its amino acid. Next up we have AAA, right? So AAA is going to have in the cell an amino acid that is linked to a tRNA molecule that is complementary to AAA, right? And that's going to come in. So for example, we have an amino acid, and there's an amino acid called lysine, so I'm just going to write that here, lysine. And so that will have a tRNA that is complementary to the next codon, right? So if this is AAA, that's going to be UUU because it's tRNA, right? So it doesn't have a T, but a U. Okay, and that's going to come in. So what will happen is that you'll have lysine here. Okay, very, very important step. Lysine here, and its tRNA is going to be complementary to the codon on the mRNA. So tRNA's codon is called an anticodon. For this reason, you don't need to know this word, but it's called an anticodon because it's complementary to the codon, right? That's a codon. So the anticodon for lysine is here, and it comes up and pairs up in the ribosome in front of its codon. bringing in the lysine. Now what the ribosome does is that once both of those amino acids, for example, are here, it's going to form a peptide bond between them. It's going to form a peptide bond between them. And you get a peptide bond between them. So we link two amino acids together. And shortly what will follow is that this mRNA is going to move along, right? So this mRNA is going to move along, and you're going to have AUG, right? And then AAA. And then a third codon is going to be here, and a third tRNA is going to come in, right? With its particular complementary anticodon. It's going to bring in another amino acid that's going to be linked with, already we have methionine, and already we have lysine, and then that's going to link up with another third amino acid, and so on. And this will keep on occurring until the protein is made. Now, this particular step in which we make the protein, just for your knowledge, is called translation, because you're translating the mRNA. So it's called translation. Well, this one here, when you're transcribing it, transcribing the DNA into mRNA, that is called transcription. Okay? And that is how proteins get made. So let me repeat this very quickly, right? Keeping everything on your screen. DNA is copied into an mRNA, a messenger RNA, in the process of transcription inside the nucleus. That mRNA then leaves the nucleus and goes into the cytoplasm where the ribosomes are. If the ribosomes read off, translate off the mRNA and use tRNA molecules to bring in, pair up the amino acids together. And then the ribosomes form peptide bonds between the amino acids. Every 3 bases is called a triplet, or a codon on the mRNA, and it codes for 1 amino acid. So for example, if you have 9 bases, then that's going to code for 3 amino acids. Now, mRNA codes, they code for each of the 20 amino acids, the codons, and here's a list of codons if you want to look at them. It's not in your syllabus, and you don't have to memorize any of these, but just for knowledge or just for being familiar with these. Basically, you have, for example, if you have UUU on an mRNA, that's going to code for an amino acid called phenylalanine, and if you also have UUC, that's going to code for phenylalanine, right? And then it would obviously bind to its complementary tRNA in the ribosome. Similarly, we looked at AUG, right? So AUG was coding for something called methionine, should be here, methionine, okay? And AA, we looked at AAA, that was coding for lysine, and so on, right? So that's how the codons work. Each of the codon codes for 1 amino acid, and that occurs in the ribosome when they pair up against the tRNA. And that step, this process is called translation. So that's how proteins get made. That's how DNA is able to help make proteins, help essentially give out the sequence of amino acids to be linked together that eventually determine a protein, and therefore will determine the function of a protein. Now that's a very big explanation, and whenever you're learning this for the first time, you want to revise this a couple of times. Please revise this, get familiar with this, because it's going to stick with you if you choose to go into a health career for the rest of your lives, okay? You're going to be using it almost every single day. You're going to be using drugs to treat patients every single day, which target these mechanisms, okay? So very, very high yield, very important information. Review this a couple of times. Shortly, very easily putting it, what a gene is, essentially, is a sequence of bases on DNA that codes for a sequence of amino acids, which eventually make a protein, which make proteins. And in each base triplet is also called a codon, and it codes for 1 amino acid. For example, we looked at AUG, that was coding for something called methionine, that's an amino acid, right? AAA was coding for lysine. So how are proteins made? Again, this is a review of what we have already finished. A gene from DNA is copied into an mRNA. The mRNA takes the message for the order of amino acids outside the nucleus to the ribosome. The ribosomes then translate this message. By the way, this step is called transcription. For AQA, you don't need to know the word transcription, but I told you this just for getting familiar with this, because hopefully you will be taking A-level biology as well. The ribosomes then translate the message, that's called translation, from the mRNA, and understand the order of amino acids to link together. Then they form the peptide bonds between them. And we've already understood in chemistry, as well as in the biological molecules, how condensation reactions end up forming peptide bonds, forming the chain of amino acids called a polypeptide chain. Once you have this polypeptide chain, kind of like this, right? So all of these amino acids linked together, it can stay like this, right? Or it can undergo further folding, like become an enzyme or something, and so on. Here's a small tip. Google the structure of insulin and have a look. Do you notice anything familiar? Maybe something familiar to this. Insulin is a protein, right? So it's going to look either like this or that. And the entire lesson really was to help you also understand mutations, right? Mutations. So what are mutations? Mutations is that you damage the DNA somehow by changing some of the bases or by deleting some of the bases and so on, so that you're able to, instead of having a particular amino acid, maybe you have some other amino acid there, right? Or maybe you have no amino acids being formed, maybe you have chain termination, you have no protein being formed. So whatever it is, something is going to come in and it's going to cause damage to the DNA. So something, maybe it could be UV rays, right? It could be X-rays, could be radioactive or nuclear radiation. Whatever it is coming in, it's going to damage the DNA. So when it damages DNA, it can damage the bases here. So let's try to damage one of the bases. Maybe we can replace this G with a U. So radiation comes in, replaces G with a U. Now we have AUU, right? AUU on the mRNA. It damages the DNA so that we get AUU, right? It comes in, damages the DNA over here so that we get AUU, right? So if we get AUU, this C becomes an A so that we get a U on the mRNA. AUU is going to code for a completely different amino acid. AUG, remember, was coding for methionine. AUU, if you notice, codes for isoleucine. So we get a completely different amino acid, which means that we're going to get a different protein, a different polypeptide. Now that protein that is different, it can either function better than expected, right? Or it can function worse than expected, or it cannot function at all, right? So any change, or it could be a silent mutation, it just functions as is, right? So a protein that does its function, for example, let's say an example of insulin, right? Even though that doesn't really occur, but let's take an example of insulin. Insulin gene gets mutated. Now insulin can either just not be formed in the first place, or it is formed but does not work, or it is formed and it works the same, or it is formed and it works much better than the previous version of insulin. So mutations can do any of those things. Let's have a look at the topic of mutations and read this through. So when a gene is used to make a protein, we say the gene is expressed, right, through transcription and translation. The physical expression of a protein, or of a gene, essentially, is called a phenotype, right? So for example, if you have a gene for blue eyes, then having blue eyes is the phenotype, while the genotype is having the gene for it. If you have the gene for curly hair, then having the gene there in the DNA is the genotype, while having curly hair actually, right, the physical manifestation of that gene is the phenotype. So for instance, we plan to make the amylase enzyme. The gene on it will be expressed to make the protein amylase. Now imagine if the sequence of bases of the DNA of amylase somehow gets altered, right? We damage the amylase enzyme. Now, a couple of things could occur. Either it could stay the same, or you could result in a different protein structure. If there is a different protein structure, maybe the resultant amylase might not have the same active site, so it might not work, right? Or by chance, the new protein structure can instead even function better. Maybe it has an even better site for starch than the previous one, right? Or by chance, the protein can instead not even be made in the first place. So there's many possibilities that occur when we have mutations, when we change the DNA structure. And so I have explained this here. What happens is, explain this here, that when we change the structure of DNA, then we get a different protein. You understand? It could get a different protein, but sometimes the proteins can also be the same. This alteration of DNA bases is called a mutation. Mutations can result in possibly no protein being made, or a protein being made that doesn't work, or even a protein being made that works better than before, right? For example, you have rabbits in a particular area. Let's draw an area here. Here's an area, and then you have rabbits that have fur, and this color is going to indicate white fur here. So, and this particular ground is brownish. Okay? So because rabbits have white fur, they're very easy to be fed up by predators. If I introduce a predator here that eats rabbits, it's very easy. Here's a predator. It's very easy for the predator to go ahead and spot white rabbits in brown color area. But if I go ahead and put brown colored rabbits here, now they're more likely to blend in. So this could result from a mutation. Maybe one of the white rabbits had a mutation that was transferred into its offspring, which gave the offspring a brown color. And then the offspring went ahead and bred and gave a lot of offsprings that have brown fur and were able to better blend in the brown environment, right? So mutations are helpful in this way because they can introduce variation which can result in maybe the organism becoming more adapted, becoming more evolved. So mutations by chance can result in possibly no protein being made, a protein being made that doesn't work, or even a protein being made that works better than before. For example, a new fur color that allows rabbits to better camouflage in the environment. Okay? The question is, what can cause mutations? What can cause mutations? We already talked about this a little bit. Mutations can occur on their own sometimes when there's errors in cell division. If cell division, there can be errors. Due to errors in cell division, which are not corrected correctly, we could have spontaneous mutations. We actually have many of these. All the time, we have some of these, at least one cancer cell a day, every single day. And these are killed by our immune system. So spontaneous mutations are very common, and our immune system keeps the cancer cells in check. We have ultraviolet light. UV light is ionizing, which means that it can even damaging it. X-rays, gamma radiation, again, go ahead and damage DNA, change the sequence of bases so that they can result in a structure of a protein, resulting in many possibilities. Chemical mutagens. Chemicals can also go ahead and cause. Now, anything that causes mutation is really called a mutagen. Okay? A mutagen is something that causes mutations, that damages the DNA. Remember teratogens? Teratogens is something that increases the chance of birth defects. Right? A mutagen is something that causes mutation in DNA. So a bit of a summary of this. Basically, we have the DNA, and DNA is a double-stranded helix, and it is made up of DNA nucleotides linked together by phosphodiester bonds. The two anti-complementary DNA strands have nitrogen bases that pair up complementarily to each other. Adenine always binds to thymine, cytosine to guanine. On the DNA surface, it contains genes. Genes are sequence of bases that code for sequences of amino acids. Those sequences on the DNA that are part of the gene, they contain triplets, right? Each of the triplets will go ahead and code for one amino acid. The triplets are called codons. The codons, DNA is copied into an mRNA that contains the codons. The mRNA goes to the ribosome, where it is translated by pairing it up in front of the transfer molecules that bring in the amino acid, right? With their anticodons. And that is called translation. We get a polypeptide chain due to peptide bond formation between amino acids in the ribosome. That is normal, but DNA can also get mutated so that you change the basis orientation in the DNA. When that occurs, that is called a mutation. Mutation can lead into a different form of an mRNA. If you have different mRNA, then you're going to have a different protein. And the different protein can either work better, not work, not being made, or doesn't have any change, noticeable change. That is called a mutation, and things that can cause mutations are these. Okay? And that is the summary of this particular lesson. So hopefully I'll see you guys in the next one, which covers a very, very high yield, very important, not just for GCSE biology, but for A levels as well as future medical students. And that is called inheritance. Now, I keep on emphasizing this to you guys, but if you have to review these videos and you have to review the notes over and over again, and you have to draw the diagrams so that the information that I'm giving you in this fast-paced environment settles into your brain, okay? And do maximum practice questions. And if you need practice guidance, and I recommend that you do get practice guidance from very experienced teachers, then you can send us a message on the WhatsApp link provided below. Alright? Like and subscribe, and please share these videos. I'll see you guys in the next one.